Okay, so today we'll be going over some recent experiments that are advancing our understanding of unified science, in particular molecular genetics. So we'll be looking at the science of evolution and development, and specifically experimental studies and quantification of hyper-intelligent evolvability and natural, non-random mutagenesis. In these unified science reviews, we often focus on experiments and theories advancing unified physics, which is a mathematical and conceptual unification of all forces, constants, properties, dynamics, and interactions of matter and energy from the quantum to the cosmological scale. But we are interested in much more than just the field of physics. One of the reasons that we refer to unified science is because we are actively extending the concept of unified physics to include a unified description of all organized matter in the universe, which extends beyond just quantum physics and relativity and into chemistry, biology, psychology, complex systems, and intelligence in the universe. So, for example, we can look at the dynamics of quantum gravity operational in the biological system, where the underlying spacetime geometry of quantum entangled wave functions of macromolecules direct the biochemical networks of the cell and the coupled information exchange of the biological system with the universal information network of space memory, where non-local signals are quasi-instantaneously exchanged regardless of distance in space or time via micro-wormholes manifesting as quantum tunneling dynamics. Our work to unified science may be a bit ahead of its time, as more conventional scientists most probably view quantum gravity and biology as two separate and distinct worlds having nothing to do with each other. Of course, this is the fallacy of a myopic and non-syncretic perspective. And time and observational science and experiment will, in time, come to show the indelible and innate link between these two domains, and indeed the innate interconnection of all domains within the universe. That's why we call it a unified science, and that's why we talk about the connected universe. In a larger purview, what we are looking at is the concept of intelligence in the universe. In the canon of conventional scientific theory, there is no innate intelligence to the universe. You have systems that replicate intelligence via mechanics. That is to say, the universe is a machine that may have functions of the machinery that seem intelligent, but it is fundamentally lifeless and blind. So within the conventional paradigm, the conventional scientific purview, the universe is like a machine, and any intelligence that might be manifest that you can see is not an innate intelligence. It's just uh, produced almost like artificial intelligence via these mechanical systems. In cases where there are observable instances of uncanny intelligence, like the biological system, the conventional scientific paradigm will evoke something like computation, i.e., the brain is computational, a machine that produces a simulacrum of intelligence. As well, there are genetic algorithms that computationally determine evolutionary traits and behaviors. But again, by equating it to a machine, a computer, you can take out the element of any kind of natural innate intelligence, and it's just will work. It's just like clockwork, a machine, lifeless and blind. So mechanics and computation generate an intelligence in the universe that is in effect superficial. That's kind of the conventional approach to explaining systems intelligence in the universe. There's strong resistance of science to accepting innate intelligence is that I believe it opens the door to perceiving natural sentience, which leads to questions of teleology. And this in turn invites philosophical or even spiritual perspectives that hard science wants to avoid or ignore. But what we'll see with some of these experiments is that we're just coming to a point in science, a point of unified science, unification of science, where you can no longer avoid or ignore uh, these critical questions of the nature of intelligence in the universe and natural sentience. So what we posit is that the universe has qualities of an innate intelligence present in the self-organizational dynamics. In fact, a hyper-intelligence and natural sentience that is not the result of mere computation or clockwork, but due to the interrelationship, often non-local, between all subsystems of the universe, which synergistically self-organize in a hyper-intelligent, fully cooperative and interconnected system. In exploration of this key concept, we will explore here the understanding that intelligence in the biological system is not restricted to a complex central nervous system like the brain. Processing of environmental information and information of internal states, generating intelligent directionality of cellular operations, resides at a much more primordial level than neuronal networks. At the heart of the cell, it resides at the heart of the cell in the genetic, morphogenic, and metabolic molecular networks of the cell, which necessarily operate at the quantum level and hence involve non-local dynamics of the wave functions. And following the postulate of ER equals EPR, Einstein-Rosen-Bridges equals EPR, non-local interactions like entanglement involve space-time geometry, quantum gravity, so it necessarily also involves the space memory network. Since the demonstrable intelligence we observe in neuronal activity evolutionarily emerges from the intelligence of the molecular networks of the cell, which possesses the same quality of information processing normally only attributed to entire neuronal networks, as a result of being a hollow fractal system with scale-free cognitive functions, we should not be surprised to find intelligence operational within the genetic machinery of the living system, which is what we'll see today in experiments and studies that are quantifying directly this trait. Because it is the morphogenic networks, the transtemporal information exchange in the space memory network that are the ultimate root of intelligence in the organism. And so this leads us to the concept of scale-free cognitive structure within the hollow fractal organization of the universe. And this is critically important to understanding the living system and what life is. In our work on the Unified Space Memory Network, one of our primary postulates is that evolution involves non-random mechanisms that have directionality as a function of system intelligence. That is, information feedback, feed-forward, and memory processes occurring within the molecular domain of the living system and within the unified physics domain as the molecular dynamics and organization are linked with the information processes of the space memory network, which gives the molecular domain of the biological system a hyper-intelligence. So an organism can direct evolutionary adaptations, not because at the organism level it is cognitively directing these changes, but because life is a nested system of cognitive domains. And that's what we're looking at here in this schematic. This is a way to kind of conceptualize this, the way that life is a gestalt of nested cognitive domains, holofractally organized. Uh, that is to say that there is scale-free cognition. We extend this across all scales. So even biochemical networks have cognitive type information processing or innate intelligence. This this is how you get like biogenesis, the emergence of life. How at one point, probably early on in the history of the universe, certain gestalts of abiotic matter transitioned to living matter. Primordial chemical networks were not blind and or random. Directional adaptation was able to proceed in an intelligent fashion to convert some biochemistry to a medium of memory with information coding and syntax, what we know today as DNA, and direct cooperative networks that formed compound intelligences like the first cell, and then multicellular organisms and onwards. So here we have a space-time depiction of what's called a cognitive light cone that defines the sentience boundary of a self for an organism. And we'll briefly examine this to get an idea of a holofractal system like the living organism, how it's comprised of nested domains of cognition. And hence there are intelligent, goal-oriented processes occurring even within the molecular domain of the cell, like within the genetic networks. So this is arbitrary cognitive individuals. So this isn't specifying any particular organism here, but this is to get an idea of what a cognitive light cone looks like. So each living system has a delimited area of concern, a region of space-time, with the organism at its center, within which its cognitive apparatus functions to take measurements and act. The borders of its cognition are schematized on a semi-quantitative state space defined as follows. The vertical axis is time. Values below the individual's now are past events, of which it may have a memory extending some duration in the past. Values above the now are future events, which it may be able to predict or anticipate to some distance in the future. The horizontal axis represents 3 dimensions of space. Each individual, based on its sensory and effector apparatus and the complexity and organization of information processing unit layers between them, can measure and attempt to modify conditions within some distance of itself. That's the body, the microenvironment, a territory, or maybe even an entire planet. The size and shape of this cognitive boundary defines the sophistication of the agent and determines the scale of its goal directedness. So we can see here non-arbitrary space-time light cones. So the cognitive sentient boundary condition of a tick is very small. It basically encompasses its body, and it does not extend very far into the past or the future. A dog has a larger cognitive light cone, as it can it's not just concerned with its kind of bodily functions, but it can actually work to influence things in its immediate environment. And ostensibly it has some extended memory into the past, and perhaps even a limited degree of ability to project or extrapolate into the future. Humans, as we know, have a pretty robust memory, so their cognitive light cone extends quite a bit into the past, and we are really adept at projecting or extrapolating into the future. So our cognitive light cone can extend quite a bit into the future. The size and shape of this Cognitive boundary defines the sophistication of the agent and determines the scale of its goal directedness. This scheme enables multiple agents, regardless of their composition, structure, or origin, evolved or engineered, to be directly plotted on the same space. So we can also make these kind of cognitive light cones, these sentience information boundaries for even things like artificial intelligence or artificial sentience. The shape of boundary defines each agent's cognitive light cone. Anything outside this region is mentally inaccessible to that system. And primitive agents, I kind of already went over some of this. The dogs have significant memory, but very limited ability to plan for the future and can only really care about events in their local vicinity. So that's where you get this shape of the cognitive light cone, for instance, for a dog. Humans exhibit a great diversity of cognitive boundary shapes, but on average have a memory that lasts approximately 100 years, can anticipate several miles away, can anticipate decades into the future, and often plan and act to attempt to modify events on quite distant spatial scales, sometimes planetary or beyond. A variety of as yet unknown alien engineered and biosynthetic life forms could occupy every conceivable corner of this option space. So that's hyper intelligence. In this scheme, individuals can overlap the same biophysical system, can support a number of coexisting coupled cells with different cognitive borders. And importantly, that's what we see here in under C is compound intelligence. This is the nested holofractal scale-free cognitive organization of the living system. Life is a holofractal nested system of cognitive domains, a dynamic evolving exemplar of scale invariant cognition in the universe. Within the cell, you have molecular domains that have an informational boundary of self and spatiotemporal cognitive light cones. These combine symbiotically into the next fractally nested level of the cell, which acts synergistically with other cells to form the organism. So here's the cognitive light cone of a cell, and you get many of these working cooperatively together that form the organism and the cognitive light cone of the organism, the sentience boundary of the organism. And then you with like social organisms, you can have the superorganism of the colony or the society. So these are fully cooperative, integrative, cognitive domains. Life is a holofractal system that is comprised of nested domains of cognition. And hence there are intelligent, goal-oriented processes occurring even within the molecular domain of the cell, like within the genetic networks. One of the key factors that we posit, and which is not depicted in this schematic, is that once you get to the molecular cognitive domain, you are at a level where quantum mechanics becomes predominant and interactions become inherently non-local. Hence, the spatial temporal cognitive light cone becomes connected with the universal field of the space memory network, and hence becomes hyperintelligent with accession to information states in all of spacetime. As you have compounding intelligences, your cognitive light cone extends so that it projects further, is able to influence further events further into the future and across larger domains of space. But as you go down to the molecular level, once you get to those non-local interactions in the quantum domain of the molecular biology of the cell, that's coupled with the space memory network. So that extends to all space-time domains. And that's why at that level, there is accession of hyper-intelligence. And that's a key point to understanding some of the experimental results that we're going to be looking at today. So here's an information processing schematic for a subcellular domain depicting a genetic algorithm with feedback feed-forward circuitry. This is an example of the cognition at the molecular genetic scale and representative of the computational boundary of cells within a biochemical network. So within the genome, you have gene regulatory networks. There's a gene regulatory network component. It has primary outputs, which are messenger RNA and the proteins that codes for. The proteins have terminal outputs, which are behaviors and structures of the cell, behaviors of the cell, structures of the organism, behaviors of the organism. And this is influenced by input signals, for instance, from the environment or from the organism. But at each step of the way, you also have feedback circuitry, feedback dynamics. That's why this is like an algorithm, a genetic regulatory network algorithm. And so depending on the behavior of the organism and its interaction with the environment, you might have a feedback signal that changes the gene regulatory network component, which changes primary outputs. So different RNA is produced, which produces different protein complements, and that in turn affects changes in cell behavior and structure. In this way, we can see where there is a degree of computational activity occurring here, and it is much like an algorithm. But one of the things that we posit is that part of this feedback circuitry, if we were to really extend the schematic, it would include information inputs, signal inputs from the space memory network, which is a hyper intelligent system, so that this gene regulatory network has a degree of innate intelligence, innate sentience that can actually produce goal-oriented behavior, not just programmed algorithmic responses. Single cells are composite agents that exhibit extremely rich patterning and behaviors and can be divided into even smaller independent units, such as cytoplasm fragments, microcondensates, and protein network subsystems. These capacities of cells presage their multicellular behavior as metazoan organisms, that is, multicellular organisms. Most features observed in the anatomical control of complex organisms, including differentiation, programmed cell death, and neural machinery, already existed in ancient unicellular life forms. Cells such as bacteria and yeast, as well as advanced plants, have been studied for their ability to compute and predict future events using patterns inferred from prior experiences. So even organisms with no neuron to speak of can show memory, learning, adaptive responses, and goal-oriented behavior, indicative of consciousness, sentience. Such neural systems have also been used to understand neural function. Thus, capacities usually assigned to entities with nervous systems, such as integrating spatiotemporal information, memory, and ability to pursue specific outcomes via selection from a number of possible behaviors. It's what I call standalone volition and goal-oriented behavior, the defining characteristic of a conscious agent. Evolved from four older pre-neural origins. The emerging field known as basal cognition tracks the evolutionary history of learning and decision-making processes, beginning from the dynamic problem-solving capacities of cellular and subcellular forms. Many examples of memory, anticipation, context-dependent decision-making, and learning are exhibited by organisms from yeast and bacteria to plants and somatic cells. This is even true of subcellular level components, for example, gene regulatory networks, the schematic of which you see here, cell signaling pathways, reaction diffusion chemistry, and metabolic networks. So the heart of molecular biology is fairly simple to understand. Deoxyribonucleic acid is a helical polymer comprised of 4 nucleobases, A, C, G, and T, that form a syntax or code that stores and carries information. For protein coding genes, that information is transcribed into the messenger molecule RNA, which specifies the linear 3D configuration of functional RNA molecules. Transcribed into molecular RNA, which specifies the linear sequence of amino acids needed to form polypeptides. DNA can store other information as well, such as the 3D configuration of functional RNA molecules like non-coding RNA, something that we will be looking at when examining the recent experimental results demonstrating non-random mutagenesis and directional adaptation. So your genome has the DNA code comprised of 4 nucleobases, which is transcribed into the RNA messenger intermediary that specifies the amino acid sequence of the polypeptide, and the polypeptide, your proteins, are the terminal products of that code, the DNA code. And these are what form the structures and signal effectors for things like behavior within the cell. One of the things is that even a single nucleotide polymorphism or variant, a single change in one of these nucleobases, can change the amino acid sequence and produce a novel or variant protein molecule, which can result in direct changes to the phenotype of the organism. So this is the core of molecular biology, and it's just important that we know this complete picture to understand some of the experimental results that we'll be looking at and their implications. So as can be seen from the transcription and translation machinery, this is what we call the fundamental 3 pillars of biology, especially molecular biology. The DNA code, the RNA polymerase, this is the RNA polymerase enzyme and resulting mRNA transcript, and the ribosome and tRNA. This is from our publication, The Unified Space Memory Network, from Cosmogenesis to Consciousness. These 3 fundamental components form the primary information system of the biological cell. In our article, we raised the question, how does such complexity within molecular biology arise? So here we have a primordial ribozyme. Theoretically, this is a molecule, an autocatalytic molecule that is able to encode its own structure and at the same time replicate itself. So it has a dual functionality. And the idea is that this differentiated into the independent functions that are now served by DNA and proteins. But this intermediary is thought to have been able to encode both of those capabilities. Um, how did this relatively simple primordial ribozyme grow to the phenomenal complexity of extant ribosomes? So this is one of the questions that we raise in our study on the unified space memory network. The configuration space for ribosomal RNA and polypeptides is nearly limitless. The number of different ways that they can adopt three-dimensional configurations. Yet there are specific few configurations that have biological utility and functionality. The configuration space is so vast that functional configurations cannot be found via a blind process of trial and error sampling of the configuration space. There must be a mechanism inducing directionality, much like the strange attractor of chaos dynamics. These kinds of considerations are what led us to postulate a non-random mechanism or non-random mechanisms underlying evolution and development, and extends the concept of evolutionary dynamics beyond just the biological, but to matter in general, leading, for instance, a simple proton electron plasma to form organic molecules that evolve into complex macromolecules that form the basis of the living system. By looking at quantum information theory and unified physics, quantum gravitational dynamics, we were able to explain how non-random evolution can occur and how it is an essential and integral component of the hyperintelligence of the universal space memory network, and even the basis for consciousness. So this is a key postulate of ours on how you can get non-random mechanisms underlying evolution and development, like non-random mutagenesis, under purely random interactions with no underlying feedback feed-forward mechanisms and with no innate intelligence or interconnectivity. The outcome you would expect for a primordial proton electron plasma in like the early state of the universe, the most probable outcome would be that it just kind of diffuses and becomes a diffuse electron proton plasma. However, what we are positing is that because of the action of the underlying connectivity network of space memory, this low order configuration of matter and energy is actually pulled more strongly into synergistic organizational arrangements like compound molecules, because as these molecular configurations grow in complexity, you get larger and larger entangled systems, entanglement networks. And following the holographic correspondence conjecture, where entanglement and these kind of non-local connections of atoms and molecules are equivalent to space-time geometry, ER equals EPR, organizations of matter that have a greater entanglement structure have as well a greater connectivity structure, connectivity network of space-time geometry, of the micro-wormhole network. So there's greater information exchange occurring here, so that actually acts as an attractor point, drawing matter into these ever-increasing levels of order and complexity. And to a smaller extent, this also occurs with non-living or abiotic matter. But there's actually a veritable entropic force emerging via transtemporal information exchange that draws matter into higher order organizational arrangements. And this can also, this is part of the way that evolution can occur in a non-random fashion, with information feedback coming through this transtemporal network of space memory. One of the key things to understanding this is the holographic nature of the universe, because from general relativity, we know that we occupy a four-dimensional space-time. However, because the universe is holographic, that higher dimension, that 4D space-time is a projection, a higher dimensional projection of a 3D information structure. The space voxels, three-dimensional voxels that encode information and memory in space, hence space memory. And so fundamentally, you have the 3D voxels of space that are coding information, and that memory is projected as a higher dimensional space-time. But that means, in the sense that the time is not linear, and it's somewhat illusionary. It's not as fundamental as space, the space voxels, and the information that they encode. So what we think of as past, future, and present, it's not a linear arrangement, and they're actually all just space-time frames that are occurring concurrently, simultaneously. So you can have this kind of transtemporal information exchange. So we posited that evolution will proceed via non-random mutagenesis within the genetic code, with purposeful or goal-oriented changes being accessed via the innate sentience of the scale-free cognitive gestalt of the organism, and via the non-local transtemporal connectivity networks of its molecular machinery with the unified space memory network. The experiment that we will be reviewing today was studying a single nucleotide variant. So first, we will review how single nucleotide changes can be generated within the genetic code via a non-random mechanism, as postulated in my paper, Unified Physics and the Entanglement Nexus of Awareness. So in the Space Memory Network paper, we described how the emergence of higher orders of organizational synergy in molecular systems will represent a higher degree of spatial-temporal entanglement of such physical systems, which through retrocausal interactions across the non-local temporal network will produce an attractor-like point towards which systems will be influenced to develop. From this, it is demonstrated how increasing complexity is, at a fundamental level, an expansion of the entanglement network of physical systems. So they're able to encode greater amounts of information into the space voxels, into space memory, and that expansion of quantum complexity of this entanglement network is directly correlated with increased connectivity of the micro-wormhole space-time spatial geometry. So it becomes more strongly connected across space and time when you have these relatively extremely complex organizations like DNA, like microtubules, and even macromolecular assemblies like the cell. Considering this, we investigate how adaptive mutations could be influenced or driven by non-local influences across the temporal entanglement network. So we can explain how you can get changes in the DNA code that affect the evolution of the organism into these more complex organizations with higher synergistic relationships. Mutations that lead to phenotypic adaptations that are beneficial for the organism will result in increased survivability, multiplication, and in many cases, increased adaptive radiation. This can be seen as an expansion of the entanglement network. When you have adaptive changes, non-random mutations in the DNA that are adaptively beneficial, the organisms are able to multiply and spread and grow. And this in turn strengthens the entanglement network and the micro-wormhole connectivity of space, making it a stronger and stronger attractor point, pulling across time to that end goal. So this can be seen as an expansion of the entanglement network within macromolecular structures such as the DNA polymer, which will result in increased retrocausal interaction influencing what would otherwise be stochastic or random changes of the DNA code and resulting phenotype to adaptively beneficial fluctuations. So how specifically would these non-local temporal interactions influence the DNA and drive supposed adaptive mutation? Although the mechanisms are probably numerous, we will investigate one such mechanism. And this is going to be important for the experiment that we'll be reviewing here in just a moment. Deoxyribonucleic acid DNA is a macromolecular polymer formed from the covalent bonding of deoxypentose nucleic acid. Okay. Deoxypentose nucleic acid purines and pyrimidines, aromatic heterocyclic organic molecules rich in nitrogen moieties, in which a sugar is linked to a nucleoside. So there are two canonical purines and two canonical pyrimidines. So your purines are like guanine and adenine, and pyrimidines are your cytosine and thymine. Those are the four nucleobases that form the genetic code of DNA. Referred to as nucleobases. These are covalently bonded together, the phosphate side chain, creating a helical filament or strand. Two such strands come together when complementary nucleobases align, so C to G, A to T, such that the purine adenine will form hydrogen bonds with the pyrimidine thymine, and the pyrimidine cytosine will form hydrogen bonds with the purine guanine. This is the normal Watson-Crick base pairing structure of DNA, forming a double helix. The heritable information of biological organisms is contained within the base pairing structure of DNA and RNA. However, while the canonical nucleobases are certainly the predominant molecular configuration, there is a dynamic equilibrium in which the canonical isomer will interconvert to a less common isoform or tautomer. This process is called tautomerization and results when a non-stationary proton within the nucleoside quantum tunnels from a common location to a less common position within the aromatic ring. And this is an example of where proton tunneling is such an important and a dominant factor in the non-local interactivity of the biological system and coupling with the space memory network and the micro-wormhole network of space memory. So when you have tautomerization and proton tunneling, you get anomalous base pairing arrangements. So cytosine, instead of in the normal Watson-Crick binding, instead of pairing with guanine, one of the protons is tunneled over to this thymine adenine, and now your cytosine pairs with adenine. If your thymine gains a proton from proton tunneling, it now pairs with guanine. This is changing the DNA code because you no longer have the fidelity that T always base pairs with an A or C with a G. But this is one of the ways that you get mutations, point-wise mutations in the DNA. When tautomerization occurs during replication, the DNA sequence will be misread and anomalous base pairing will occur, as I explained. This is showing of the quantum tunneling dynamics, where that normal Watson-Crick base pairing and normal isomer configuration of the nucleobase is energetically favorable. So it likes to be in that state. It's thermodynamically stable and at a low energy value where it likes to hang out, so to speak. And normally it would not cross over into these less common isoforms, the tautomers. But because we're talking about quantum mechanics and things are occurring in a non-local fashion, the protons that form the hydrogen bonding pattern of the DNA code those protons can tunnel across that energetic barrier almost for free and adopt these less common isoforms. Now, keep in mind that tunneling, how is that physically occurring? It's occurring via the micro-wormhole structure of space memory. When this proton tunneling is occurring, you're having information exchange with the universal information field of space memory via the quantum space-time geometry. Since such quantum tunneling of non-stationary protons on the nucleobases is constantly occurring, the nucleobases can be thought of as existing in a superposition between multiple isoforms. As occurs in decoherence when DNA interacts with the environment, which is to say that when information from the environment is transferred upon the DNA, becoming entangled with its surroundings, it will collapse the nucleobase superposition, either as one of the canonical bases or as a tautomer. Since the bases will be more strongly correlated through the entanglement nexus with sequences that promote greater survivability and adaptation. Remember, going back to figure 1, that connectivity network of space memory, that will promote greater survivability. It will preferentially select a tautomer that drives adaptive mutation. So information across that space memory network, when this is occurring and you have that coupling with the micro-wormhole structure, it will preferentially select a tautomer that drives adaptive mutation because the end result of that is a expansion of the entanglement network. And so a greater entropic force, a greater ordering force towards that kind of synergistic arrangement, organization. This is one such way that the entanglement nexus will function in natural selection, evolution, and development, since this non-local temporal interactivity will drive self-organizing systems to higher synergetic ordering and convergent complexity. And we'll see this convergent complexity in a study where this is observed. It will be a fundamental characteristic of the complex macromolecular systems of the biological organism. As such, it may be an integral mechanism and engendering protosentience and self-organizing adaptive systems, expressing increasing levels of awareness within the biological organism. I'm going over this before we get into the experimental studies so that you have a baseline understanding of how we can explain what these experiments are finding, the results of some of these studies. And just, an important part of this proton tunneling is that your normal ATGC code, nucleobase pairing, when you have this tautomerization, that GC is flipped to an AT. So it changes the genetic code. Your AT, when you have the tautomerization, is flipped to a GC. So it changes in a pointwise fashion the DNA code.